Hi, I'm Stuart Brown, a faculty member in the Center for Health Informatics and Bioinformatics. And this is our CHIBI faculty seminar series, last one of the year. And today I'm going to talk about some work that we did on microbial genome sequencing and some software that we built to make it easier to make comparisons between genomes of microbial strains. I'm going to start out by saying this was a collaboration with the NYU College of Dentistry with the lab of Paige Caulfield and postdoctoral fellow and then research scientist Sylvia Argyman led the project. And she isolated the samples, and we worked together on a lot of the informatics. Kranti Kanganti, who's no longer with us, did the initial shell scripting and programming and blast searching. And then most of the software that I'm going to show here was written by Hao Chen, who's still a research scientist or bioinformatics programmer in the CHIBI. And also Alex Alexienko helped me with some of the bioinformatics. This was supported by a CTSI grant to Dr. Argyman, and thanks to Konstantin Aliferis for support at CHIBI and the High Performance Computing Facility, which provided the compute power to do the assemblies and everything else, really. So that's Kranti, that's Hao Chen. Okay, so this is a story about tooth decay. Severe early childhood caries, they call SECC, is a severe tooth decay that's found in children ages 2 to 5, high prevalence among low-income children, and it presents like this with extreme enamel hypoplasia. And malnutrition is a major cause. The association between Streptococcus mutans and caries is very striking in the severe early childhood, with some studies reporting that S. mutans levels represent as much as 40% of the cultivable bacteria that can be harvested from the mouth in children with this disease. So this is Streptococcus mutans. It's a facultatively anaerobic gram-positive bacteria found in the human mouth. So an important fact is that Huck's postulates have not been fully satisfied for this organism as the cause of tooth decay, because it's found both in the healthy mouth and in cases of severe tooth decay. However, the prevailing wisdom is that S. mutans is the main contributor to tooth decay. And there have been a variety of hypotheses that there are specific pathogenicity factors involved in the strains that are found in the case of severe tooth decay versus the bacteria that are isolated from a healthy mouth. And so that was our goal for this project, is to try and find out if we could isolate genetic differences between the SECC strains and strains that were isolated from healthy mouths of children. We know that there's great phenotypic diversity among S. mutans strains, a wide variety of different microbiological parameters, different genetic markers, etc. Many years of prior work by many investigators in other bacterial systems point to pathogenicity factors, insertion sequences, unique genetic elements that confer pathogenicity. And so that's what we were looking for. This work is following up on some prior work in the lab of Paige Caulfield, where they had done subtractive hybridization, in test tubes to try and isolate chunks of DNA that were unique to the pathogenic strains. So our goal was to use a genome sequencing approach to find unique genetic elements in the S. mutans strains isolated from the SECC patients. So that was our basic plan for the project. DNA sequencing has become so inexpensive over the course of this project that our plans to do sort of complicated sequencing based on DNA subtraction changed to let's just sequence a whole bunch of strains completely and compare them computationally rather than trying to filter elements that are in the pathogenicity strains and not in the healthy strains. So we used paired end, 100 base reads, so 200 bases, 100 from each end on an Illumina machine, which generates, in the time that we did this, about 40 gigabases of data from one lane. We could put 20 bacterial strains in that one lane and get 2 gigabases of data, 2 billion bases per strain. Since the genome size of strep mutants is about 2 million bases, that's 1,000-fold coverage for each strain, all for the price of one lane of sequencing. It's really a game changer in microbial biology to be able to take a whole collection of strains and sequence them for in the neighborhood of $1,000 or so. The concept of subtractive hybridization is a well-established molecular biology technique. The idea is that the DNA in one tube has some sequence elements that are missing from another tube, and so you can subtract them, and whatever is left behind is the unique elements. And there have been a variety of different molecular strategies to do this. Usually you work with relatively small pieces. We did this project using restriction fragments in the past that were 1,000 to 500 bases in size. And so our goal was to sort of emulate that type of experiment computationally to develop an in silico subtraction method. So we had 10 single colony strep mutans isolates from these severe Caries patients and 10 single colony strep mutans isolates from healthy patients. I should point out that prior work in Paige Caulfield's lab had shown that in general, in each patient, you have just one dominant strain that makes up more than 90% of the isolates that you find. So just sequencing one isolate from each patient, we believe that that gives us a valid picture of what's going on in the strep population in that mouth.
Stuart, this 10 single colony from 10 patients?
Yes. Okay. 10 single colony isolates from 10 different SEC patients. Yes, not all from one for sure. So the idea here is to find the fragments in one genome that show no similarity, and we used BLAST, which is a common similarity matching tool, to another genome or a group of genomes. We're comparing the whole genomes, looking for genomic regions unique to each SEC strain and not in any of the Caries-free, that's the CF, Caries-free strains. We also did the reverse search, looking for sort of non-pathogenicity sequences, sequences that might block infectivity. So we looked at all the strains from the Caries-free patients, subtracted out the SEC genomes. We cut the test query genome into small fragments. We did 500 bases, and then we went back and did 200 bases to see if we missed anything. We created a custom database and set our threshold for no similarity at a BLAST e-value of e to the minus 12. And I'll show you why we chose that in a second. So just as a graphic for the general approach here, so we sequenced these 10 genomes and independently assembled each one, and sequenced these 10 genomes and independently assembled each one. Took all of them and chopped them up into small fragments, and then built a database out of these fragments and a database out of these fragments. And we did a BLAST of each one of these against all of those, and each one of these against all of those. And that gave us the fragments that were present only here and the fragments that were present only here. Then we compared all of these against each other to find fragments that were common in the pathogenic strains and fragments that were common in the non-pathogenic strains. So these would be, ideally what we were looking for was, the pathogenicity factor that would be present in all of these and none of those. Or similarly, something present in all or many of these and none of those. Yep. So pathogenicity associated sequences, that was our goal. So when we did the 1,000-fold coverage, we got a single FASTQ file from each strain, and we used the software called Abyss to do de novo assembly on each of the individual genome sequences. So we did it 20 different times, optimizing each one. One of the reasons we did this rather than map each strain against a single reference genome is that we were most interested in finding the genomic regions that were not part of the reference genome. And those would have been unmapped sequences. And also, there's quite a lot of genomic rearrangement in strep mutans. So since we were able to cost effectively get very deep coverage, we decided to go for a de novo genome assembly. Doing 20 whole genome assemblies used a significant amount of CPU power and RAM from the high performance computing facility. So we thank Stratos and Eric for that. Once the genome assemblies were completed, we compared the size and GC content of each genome to the reference, and we found that the size was very similar. You know, there's some variance. Many of the de novo assembled genomes were a little bigger than the reference due to the fact that we found novel insertion sequences in each one. And the GC percent of the assembled genomes is very similar to the existing S mutans reference genomes, of which there are a few. This is just a table of some of our assembly statistics. The GC percent doesn't vary very much between the different strains. The N50s, which is a measure of the average contig size, actually it's the size of the fragment that contains 50% or more of the assembled genome. And these are big numbers, 80,000, 90,000, 120,000. So we're not getting one perfect single chromosome, but we're getting very big chunks, and most of the genome is in those very big chunks. And the total number of chunks that's left is pretty small. So there's a couple of big ones and a small collection of smaller ones. It's very hard, even with 1,000-fold coverage, to get a perfect single chromosome, a single read-through of an assembled genome. There tend to be some repeats that are hard to assemble. And it requires a certain amount of finishing, which usually means identifying fragment ends, doing PCR, building clone libraries. And we didn't want to do that on all 20 strains. We just checked that we had the genome present with very small, well, very little missing. And we concatenated those 100 or so fragments from each strain into a single multi-sequence FASTA file and called it a draft genome. We took those draft genomes and we aligned them to the reference genome. This is using a software called the MAUVE Contig Mover. And what we saw was that there's good continuity, although sometimes a bit of rearrangement of chunks, but they're big chunks. And then in each of our sequenced genomes, there's usually a little bit of content somewhere along that doesn't match back to the reference genome. Those are the inserted sequences. And so we did a check like this for all of the 20 strains, and they all look like they have very good assemblies. Then we had to set a threshold for declaring fragments not similar, which is not entirely trivial. BLAST has an e-value statistic which is based on a probability which is not strictly the same for a longer fragment versus a shorter fragment. So you can get a BLAST expectation value of e to the minus 12 on a shorter sequence like this one that has just a single mismatch but over 39 bases, or the same e-value for a longer sequence, 99 bases, that's 84% identical. So we did a little simulations and a little bit of testing, and what we found was that the number of unique fragments, the number of unmatched fragments between each genome and the reference sequence did not change much across a very wide range of e-values. So the results of our analysis were not sensitive to where we set the e-value threshold. We were relatively stringent. We set it at e to the minus 12. But going back and repeating the analysis many times with different e-values didn't really change the number of fragments that we found at the end. So that made us more confident that this analysis was going to work and not generate a lot of artifacts. At about this time, when we were doing lots of different comparisons and changing the fragment sizes and the e-values, we realized that the collection of hand-built scripts that we were using wasn't very user-friendly. It was built by Kranti, and basically only he could use it because it was a collection of different scripts that were in his personal directory. And we thought about writing some instructions and passing it around to other people in our group who were working on this. But we also realized that this idea of sequencing many genomes and comparing them to each other in groups was catching on. This is something that microbiologists might want to do quite frequently now that it's cheap and lots of people have strain collections and phenotypes that they might want to test for. So we decided it was time to build a software toolkit that would be accessible for everyone, for every lab biologist. And because these genomes are pretty small, after the assembly, the blast comparisons don't require a whole lot of CPU power and RAM. You can do these comparisons on any laptop, given a software toolkit that makes it easy. So we decided to build this toolkit, and Hao Chen joined our group and did the programming in Java to make a piece of software that would be easy to use on many different operating systems. So the product of that is something that we call the MGC tool, Microbial Genome Comparisons. And the only prerequisite to run this thing on your computer is that you have to also have the NCBI BLAST software installed, which is very easy to set up on Windows, Linux, and Macs. So I don't think that's too high a burden to ask. And we didn't feel right in trying to redistribute NCBI BLAST as part of our own software. It's like, go to their website, download the thing, hit the install button, and you're done. Another nice feature of this tool is that it has a project management layer. So you can do many different sets of comparisons with many different sets of data, and it creates a set of subfolders and organizes them as an MGC project. So when we were doing things at different thresholds or validating against different reference genomes, we were able to set up a bunch of different projects and keep our data well organized. Simple interface. You have a set of draft genomes or reference genomes or any other sequence in a FASTA file format, and you just locate it on your hard drive and you load it into the project. You can load as many FASTA genomes as you want and name them anything you want, and it'll keep those file names, or you can rename them as you go. Then you can create BLAST databases, custom BLAST databases on your own computer just by selecting among the genomes that you've loaded. So you can make a BLAST database of one reference genome or a BLAST database of as many genomes as you want as a comparator. So you're going to test genomes, a set of genomes X against another set of genomes Y. Each one of those can be made into its own BLAST database with just one click. So it sort of saves the command line work of building databases using the BLAST toolkit. Other features of the MGC program, you can break the query genome into fragment sizes, any size you like. We used 200 and 500 base pair sizes because those worked well for us. We were looking for a piece of DNA that was big enough to code for something functional. We weren't interested in unique sequences of only 20 or 30 bases, and we weren't interested in really big pieces because we might miss something. So you can find non-matching fragments in each individual genome by testing genomes one by one against the BLAST database, or you could make a query genome that consists of many strains and test them all at once against the BLAST database. Doing it that way, having a group of queries versus a BLAST database with a group of sequences, it also has a built-in tool to find the shared fragments that are non-matching. So it tests each genome, finds the non-matching fragments, and then tests them against each other to say which of the strains have the same fragment in your query that's not found in your test database. The output files are stored both as tab-delimited tables that list the fragment and its location on the query genome, and as actual BLAST reports. If you like to look at the alignment and double check, the BLAST output files are stored in their native format. Another thing the tool does, which we found that before we had the tool, we found we were doing by hand, is very frequently you get an insertion sequence that's quite a bit bigger than the fragment sizes we were testing. And this joins adjacent non-matching fragments together to form what we've been calling an element. So this is a chunk of DNA in one strain that's not present in our reference, or the group of database genomes that we're using as a reference. Another tool that we threw in, or another function that we threw in, this one was for Marty Blazer, was to do a self-comparison of one genome against itself to find sequences that were repeated. So you have a genome, you break it into small fragments, you blast each small fragment against the genome, and you record all those fragments that match in more than one place. And you get a list of how many times each fragment matches. So it's a nice tool for finding repeat sequences. There's other tools for finding repeats, but since we had the architecture for doing these blasts against genomes, it was very easy to add that tool. Then once you have a set of fragments that you found interesting, they're in the query genome and not in the target genomes, then you could send those fragments by the remote BLAST feature to the NCBI BLAST server and get annotation information. You can search bacterial or just the general non-redundant database it's a translated search. So you're translating your DNA fragment and comparing to the protein database. And in that way, we were able to identify the majority of fragments that we isolated using this method. So getting back to the, last thing is this software is now available as a SourceForge project, Microbial Genome Comparison. So if anybody wants it, you can download it there. If it doesn't work nicely for you, you can email Hao Chen, who's sitting right there. Now getting back to the Strep mutans project, using this tool, we found a total of 254 fragments that were unique to the SECC strains not present in the strains from the healthy mouth, from the caries free. Out of these, those are actually joined fragments. Those are elements, not individual 200 base pieces. There were way more than 1,000 small ones. 187 of those 254 were unique to just one strain. So they were strain-specific insertions, and therefore we kind of ruled them out as being general pathogenicity elements. 67 were shared by at least 2, but the maximum number of strains that shared any one fragment was 4 strains out of the total 10. So we found no fragments that were not in the healthy mouth and present in many or most of the pathogenic strains. So that kind of was a negative proof of the hypothesis that there was a pathogenicity element responsible for the S mutans in the mouths of the children with severe tooth decay. the reverse, interestingly, there were a larger number of these unique fragments in the strains from the Caries free mouth. And 827 of those were single strain specific. And again, no single element was present in more than half of the Caries free isolates. What we learn is that there's a lot of unique genetic elements in strep mutans, which I guess we already knew, and that there was no single element that is unique to the Caries causing or the Caries free strains. Right, many of the adjacent fragments were located in contiguous blocks. Each block was from 1 to 39 kb in size. So 39 kb is a pretty big insertion in a bacterial genome. They contained from 1 to 20 open reading frames. In the SECC specific fragments, 48 elements, 41 were found in only one strain, 99 strain specific. Okay. We found unshared elements, no generally right. Said that already. The functional characterization, no common no virulence elements were found. Mostly these were transposon associated and viral associated sequences, insertion sequences. Sylvia did this characterization using the GO ontology for all of the fragments that we found. So a few of these secondary metabolism. The replication strand associated are mostly transposon type, transposase type genes. A few other assorted things that were kind of interesting. So that was our characterization of the functions. Just because this was sort of a negative result, we also tried to do a positive control. We went and grabbed some strains that had been previously published associated with Neisseria meningitis, 3 pathogenic genomes, and 3 non-virulent strains called Caries strains. And this was described in this paper by Sean in 2008. Using the MGC on these genomes, we found a total of 103 elements that were shared by all 3 pathogenic strains and not found in any of the non-virulent strains. So a very different result than we found with strep mutans. And when we characterized those pathogenic elements, we found 8 previously identified genes. A lot of them were from the prophage NF1 and in the FHA locus. And we found 1 additional NF1 gene that was not previously reported. So our method was more sensitive than the work they did in 2008.
Mm-hmm. The fact that you picked a colony from each patient, okay, wouldn't that possibly introduce some sort of a bias that you may have, I don't know, but this strain which goes faster than the others? Or
well, prior to this, in the work where they did the subtractive hybridization, they initially picked 20 colonies from each patient, and they compared them to each other, and they found, 95% of the time that they were genetically identical. So it doesn't preclude the fact that there might be another strain that grows less well, but the general notion is that each patient has one strain, one major strain. And in fact, Paige has done some phylogenetic work indicating that you usually get your strain from your mother. People kiss babies, stuff like that. So, and they're persistent over the life of the patient. So that just seems to be the case for strep mutans. It's not a very diverse ecosystem. So this is the publication on the strep mutans work, which has now been accepted in Infection, Genetics, and Evolution. And this is the manuscript which is submitted under review in bioinformatics on the MGC tool. And that's all I have. Yes?
So I don't know anything about how big the bacterial load is in a mouth, whether healthy or with cavities. But do you think it would be possible to do the same sequencing without growing the bacteria to perhaps avoid the possible issues with some, you know, particular strains or something?
Well, that would be
not growing that well.
That would be a whole genome metagenomic project, because you'd probably get more than just the strep. So yes, you could do it, but not for $1,000 per patient. Not for $1,000 for 20 patients.
Right, and you would have to, I mean, I guess sequence deeper because you will have human DNA. That
Right, you'd have to subtract the human DNA and then have enough to assemble whole genomes for all of the different microflora present in the mouth. And there are projects like that, but this was a project about strep mutans as the proposed cause of tooth decay, because that's the focus of the Paige Caulfield lab. And so, and they had done all this prior work showing that there was one dominant strain in the mouth of strep mutans.
That was my save.
So absolutely we could do that, but not for the kind of money that we were spending on this project.
But as the price of sequencing is going down and also the read length is a
Yep.
bit higher, it may be something to consider.
Well, absolutely there are metagenomics on the mouth projects, but I don't think this approach would work very well, because you don't have nearly the level of confidence that you've got everything there. When you're looking at one colony and 1,000-fold coverage, you could start to say, you know, if something isn't there, then it's not there, as opposed to, well, you know, we did a metagenomics assembly of 40 or 100 different genomes isolated from this one mouth, and we don't find this particular 1 kb fragment in that metagenomic assembly. Therefore, we have what level of confidence that that is really not present.
But perhaps, I don't know, I was thinking in terms of maybe an interaction between different bacteria, so maybe there's something about S mutants growing with something else that makes it, I don't know, maybe
Great.
from biofilm or whatever it is that
Great
bacteria
grant
do.
proposal for a metagenomics project. I'll get Sylvia right on it.
Be happy to write the sequencing.
Right. Yes, Mickey.
Sure. Is the diversity of the strep mutant species within the Caries-free population larger than, it seems to me from the, some
Seems
of
to
the
be
numbers
yes.
you brought.
Although we probably should have done an alpha diversity type of analysis, either on those fragments or just on the genome in general, like maybe doing an alignment and looking at the level of sequence variance among the different strains. But it seemed to be, yes, that we were seeing more diversity in the healthy mouth. All right then, thank you.